Hello, I'm Bjarne Bartlett. I'm a doctoral student in molecular biosciences and bioengineering. Um, I work with Dr. Mikey Cantar, who's my advisor, and I'm really excited to present some genotyping and biomarker analysis that we did for Hawaiian Taro, trying to look at developing some new tests to determine different lines of tarot. And I'm very happy to be here today. So thank you for the opportunity to present some of what we did. Marker assisted selection. This is a strategy that's gaining importance in plant breeding. It has some potential to improve the efficiency of the breeding process, and this is specifically through the ability to precisely identify the transfer of relevant genomic information in different lines of organisms. Breeding is often based around qualitative traits. Genomic markers allow the prediction of these types of qualitative traits. You can see kind of on the left an example of a trait that's you see an example of a trait that has very discrete values, like so flower color like red, blue, purple, and then you can see a trait on the right that has a more continuous spectrum of value, like yield. And each of these traits has different genomic characteristics kind of underlying it often. And breeding attempts to select for many a whole spectrum of different traits. So the biomarkers can really help narrow down what is going on in the plant breeding process. Plant breeding itself, it's important to say, has the potential to enrich populations for these beneficial traits that have a particular flower color or improved yield. But it's not actually a tool to create beneficial traits. It selects for them, and biomarkers have the potential to make the process more efficient. Various problems with selection necessitate the need for biomarkers. A genetic marker is a Mendelian gene that's easily evaluated, or other polymorphism, right? Not necessarily a gene, closely linked with a gene, for which selection is difficult but desirable. So these genetic markers kind of identify where selection can be valuable. Genetic markers benefit selection in multiple ways. In the low heritability category, where the phenotype isn't so easily linked to a genotype, where selection requires large populations and multiple trials to reveal beneficial traits, biomarkers can help make that process more efficient. They can also be useful for selecting mature traits in mature plants at the seedling stage. This often occurs, mature traits occur later in the growth cycle, so this can reduce the amount of time that study would take. Biomarkers can be useful for selecting disease resistance in the absence of disease, and they can also be useful for selecting recessive traits in segregating populations that must self-pollinate to identify recessive homozygotes. These are all situations where finding a genomic marker for a trait that's being selected for can improve the efficiency and ability to do breeding and selection. So what makes a good biomarker? So two different traits often make a good biomarker. These are sensitivity and specificity are something we often talk about. And this means specific to the trait that's being specific to the beneficial trait, right? Whether that be a red flower or specific to a blue flower or specific to a particular other characteristic, right? So you have specificity and then you have sensitivity. So sensitivity as opposed to specificity means, sensitivity means how many of the plants with that particular biomarker have red flowers, right? So we want it to be both sensitive and specific to make a good biomarker. Additionally, inherited simply where the results are unambiguous without environmental influence. Displaying codominance where heterozygotes can be distinguished from homozygotes, where the marker's plentiful in the genome and distributed randomly, and having stable genomic features with wide applicability across many populations within the species. We know species have, populations in different regions or different genomic subsets, as well as easy to evaluate, right? This means cheap and quick, and also not requiring very specific tissue for expression, expressed in many different tissues, able to be evaluated that way. So all these make a good biomarker. So molecular markers have a lot of value in plant breeding. They can speed up the process of selection, by which I mean selection for specific genotypes and traits in plants that are being breeding is being done for, as well as decreasing the breeding cycle time and allowing for selection on multiple traits. So Taro specifically has genomic resources available recently. Chromosome level assemblies that have recently been released have provided new insights and resources available for scientists studying Taro. These published data sets allow us to compare different populations. So this is really, hopefully able to shed some light, on the differences between Taro populations that haven't been seen before. So we sought to identify genomic resources that were specific to the UH breeding program. Luckily, genotyping has been performed, and the raw sequencing data from existing populations that are specific to the UH breeding program, these can be combined and looked at together. So we're grateful for that. So we had access to that data to look at, to do some biomarker analysis. We acquired the data that we used from the SRA database, as well as in-house sequencing data from 15 newly developed Taro cultivars. So we have 93 different Hawaiian Taro lines. This came from the SRA database. And the project number you see is listed there. So these are Hawaiian Taro, and then the newly developed cultivars. These were not yet in the SRA database. These were sequenced on a HiSeq 2500. And these 15 new cultivars that we had the HiSeq 2500 data from were from the UH breeding program. So with this data, we first used FastQC to look at the baseline quality of the FastQ files. And then we trimmed the adapter content from the demultiplex samples. We used Trim-O-Matic for that. Across 108 samples, we found 287 million reads total. These sequences were mapped to the Yin et al. Cultivar from just this past year. And we used BWA and the MEM algorithm for that. And we called Variance next. This we used VCF tools and pilot, and we filtered with VCF tools. So once we had the variance, we went about looking at how to make a DNA fingerprint to distinguish the different types of cultivars. We started with this program called VAST. We were able to run VAST on multiple different merged VCF files with the following criteria to find regions of divergence between the different germplasm pools. Graphical genotypes were constructed using the software Tassel. And then after the alignment, we had 143,000 SNPs in 108 different lines. And after filtering for quality, we ended up with 3,500 SNP sites. And then we filtered them manually. Those 3,500 down to 37 sites in the 108 lines. And out of the 37 sites, we can use 8 of those to distinguish between the new lines and the Hawaiian lines. So we're really looking at 8 SNP sites between as our, our potential biomarker. And so first we wanted to see whether we could differentiate the different germplasm pools this way. We did principal component analysis among the 105 different cultivars using our SNPs, and the principal components were derived from the SNP set in Tassel. This was filtered for heterogeneity and presence of useful data in a sufficient number of cultivars sequenced. So of our 143,000 SNP sites, 352 of them were used to do the PCA analysis. The PCA analysis of these 352 sites can clearly distinguish between the Hawaiian taro in the yellow you see and the new taro cultivars in the green you can see there. So this was encouraging. So we went about trying to make a SNP profile with fewer markers. And this is a heat map of the 37 sites that we filtered down to in the 105 taro cultivars. Of the 37 sites, 8 were used that could differentiate between the 15 elite breeding lines from the University of Hawaii and the 90 other lines based on the SNPs observed. The major allele sites in this heat map are in orange, and the heterozygous sites are in blue. Sites that cannot be resolved, these are in white. And so you can see in the boxes the 8 sites that we can use to distinguish between our lines. You can see the separation between the other taro lines and the breeding lines from UH. So this was our kind of SNP panel that we can use to distinguish between these two populations. So in conclusion, combining historic data with new data is possible for the purposes of studying many things, including developing genomic biomarkers. Distinguishing between the germplasm pools of the taro, between the new elite breeding lines and the existing UH lines is possible. 8 biomarkers were sufficient to distinguish the new breeding material from the traditional Hawaiian cultivars. And so this was encouraging for the biomarker. I'd like to open this up to questions on how this was done. I'd like to thank Susan, as well as everybody who's attending today, who assisted in organizing this conference. I think it's really wonderful. And I'd like to thank my advisor, Mikey Cantar from Tropical Plant and Soil Sciences. He's my mentor and advisor. And the summer student as well. We had a summer student assist us with this project, an undergraduate student from Gettysburg College who did a wonderful job by the name of Carter Zamora. He came here with the REU program, so we're really grateful to Carter. If you have, okay, so type your questions into the chat box. They'll be answered during the question and answer session later. And the next speaker needs no introduction, but is Mike Shintaku, and I'll let him take it away and introduce himself. Thank you so much for your time.